Hi dear students, welcome to our session on Mycobacterium tuberculosis. In 1882, microbiologist Robert Koch discovered the tubercle bacillus at a time when 1 of every 7 deaths in Europe was caused by tuberculosis. He isolated the mammalian tubercle bacilli on heat coagulated bovine serum and proved its causative role in tuberculosis. And for this discovery, he received the Nobel Prize in Physiology and Medicine in 1905. Let's see the general classification of mycobacteria. The first group is the tubercle bacilli, and in that, Mycobacterium tuberculosis causes tuberculosis in human beings. And other species include Mycobacterium bovis, which is causing the cattle, and Mycobacterium microti, causing the rodents, that is murine, and Mycobacterium avium, related to the birds, and Mycobacterium marinum, infecting the cold-blooded animals. And the second group is the leprae bacilli, which is causing leprosy in humans, and it is caused by the Mycobacterium leprae. And in rat, it is caused by Mycobacterium leprae. The mycobacteria causing skin ulcers include Mycobacterium ulcerans, Mycobacterium bellni. And the other group is the atypical mycobacteria, or the Runnian group. It includes photochromogens, cotochromogens, non-photochromogens, and the rabbit growers. And the other group is the Jones bacillus, that include the Mycobacterium paratuberculosis. It usually affects the small intestine of ruminant animals. And the next group consists of the saprophytic mycobacteria, that includes Mycobacterium bupricum, Flay, Styrocoralis, Magmatis. Mycobacteria are slender rods, and they sometimes show branching filamentous forms resembling the fungal mycelium. They are aerobic, non-motile, non-capsulated, and non-sporing, and they usually have a slow growth, and they are usually acid-fast bacilli. It is an obligate aerobe, growing most successfully in tissues with a high oxygen content, usually the upper lobe of the lung and the kidney. Its cell wall contains several complex lipids that include long-chain fatty acids called mycolic acid. It is relatively resistant to acids and alkalis. Sodium hydroxide is used to concentrate clinical specimens, as it destroys unwanted bacteria, human cells, and mucus, but not the organisms. They are facultative intracellular pathogens, usually infecting mononuclear phagocytes like macrophages. Mycobacterium tuberculosis are usually long, slender, straight, or slightly curved. And they are intracellular, and they consist of mycolic acid, waxes, and lipids in the cell wall. The high lipid content of their cell walls make the mycobacteria acid-fast and hydrophobic. Because the cells are hydrophobic and tend to clump together, they are impermeable to the usual stains like the gram stain, so they are neither gram-positive nor gram-negative. They are known as acid-fast bacilli because of their lipid-rich cell walls, which are relatively impermeable to various basic dyes unless the dyes are combined with phenol. Once stained, the cells resist decolorization with acidified organic solvents and are therefore called acid-fast. Other bacteria which also contain mycolic acid, such as Nocardia, can also exhibit this feature. When stained with carbonyl fusion by the Seale-Nielsen method or by fluorescent dyes like rhodamine or Oramin O, they resist decolorization by 20% sulfuric acid and acylic alcohol for 10 minutes. The reason for acid and alcohol fastness is due to the presence of the bacillus of an unsaponifiable wax or to a semi-permeable membrane around the cell. It is related to the integrity of the cell and appears to be a property of the lipid-rich waxy cell wall. Staining may be uniform or granular. Once stained, the acid-fast bacilli can be seen as pink or red color. They appear as clumps, pairs, or long filamentous forms, and the other cells will take up the color of the counter stain, and the counter stains used are methylene blue and the malachite green. Coming to the cultural characteristics of Mycobacterium tuberculous, the bacilli grows slowly and the generation time in vitro is being around 14 to 15 hours. The colonies appear in about 2 weeks and may sometimes take up to 88 weeks. And the optimum temperature is 37 degrees Celsius and growth does not occur below 25 degrees Celsius or above 40 degrees Celsius. The optimum pH is 6.4 to 7. Mycobacterium tuberculosis is an obligate aerobe, while Mycobacterium bovis is microaerophilic on primary isolation and it becoming aerobic on subculture. Several media, both solid and liquid media, have been described for the cultivation of tubercle bacilli. The solid media contain egg, the Lausten-Jensen, Petragnini medium and the Dorset medium, and the blood containing Tarsch's medium, serum containing Loeffler's serum slop, and potato containing Pawlowski's medium. The solid medium most widely employed for routine culture is the Lausten-Jensen medium, and it consists of coagulated hen's egg, mineral salt solution, asparagine, and malachite green. And among the several liquid media, it includes Tubos medium, Middlebrooks medium, Prosper and Beck's, Sulas, and Sorton's medium. Liquid media are not generally employed for routine cultivation but are used for sensitivity testing, chemical analysis, and preparation of antigens and vaccines. The ingredients in Lausten-Jensen medium, that includes coagulated hen's egg, it will neutralize the fatty acid, the glycerol is acting as a carbon source and the asparagines will be acting as a nitrogen source, and the malachite green is present to inhibit the growth of other bacteria. The growth in liquid media appears in the form of pellicles, whereas in Dubose media, there is a diffused growth can be observed. Also, the virulent strain will be having a dispersed growth, whereas the virulent strain will be having a serpentine coat-like appearance. Tubercle bacilli also grow in chick embryo and tissue culture. Mycobacteria are not specifically heat resistant, and it is being killed at 60 degrees Celsius in 15 to 20 minutes exposure. Survival is influenced by the material in which the bacteria are present. Cultures may be killed by exposure to direct sunlight for 2 hours, but bacilli in sputum may remain alive for 20 to 30 hours. They are relatively resistant to disinfectants and can survive exposure to 5% phenol, 15% sulfuric acid, 3% nitric acid, 5% oxalic acid, and 4% sodium hydroxide. Ethanol is a suitable disinfectant for skin, gloves, and clinical thermometers. They are sensitive to formaldehyde and glutaraldehyde. They are destroyed by tincture of iodine in 5 minutes. Several biochemical tests have been described for the identification of mycobacterial species. The first one is the niacin test. Human tubercle bacilli form niacin when grown on an egg medium. When 10% cyanogen bromide and 4% aniline in 96% ethanol are added to a suspension of the culture, a canary yellow color indicates a positive reaction. The test is positive with human type and negative with bovine type of bacillus. The next test is the aryl sulfatase test. This test is positive only with the atypical mycobacteria. The bacilli are grown in a medium containing 0.001 molar tripotassium phenolphthalein disulfate and 2 normal NaOH is added drop by drop to the culture. A pink color indicates a positive reaction. Next is the catalase peroxidase test. In this, a mixture of equal volume of hydrogen peroxide and 0.2% catechol in distilled water is added to 5 ml of the test culture and it allowed to stand for few minutes. Effervescence indicates catalase production and browning indicates peroxidase activity. The atypical Mycobacteria shows catalase positive and peroxidase negative, whereas the Mycobacterium tuberculosis is catalase negative and peroxidase positive. Next is a nitrate reduction test. In this, sulfanilamide and N-naphthyl ethylene is added to bacterial suspension in nitrate medium. A positive reaction is indicated by red color, and this test is positive for Mycobacterium tuberculosis, whereas negative for Mycobacterium bovis. The next biochemical test is the amidase test. Here, the ability to split amides has been used to differentiate Mycobacteria. A useful pattern is provided by testing 5 amides, namely acetamide, benzamide, carbamide, nicotinamide, and pyrazinamide. A 0.00165 molar solution of the amide is incubated with the bacillary suspension at 37 degrees Celsius, and 0.1 ml of magnesium sulfate, 1 ml of phenol solution, and 0.5 ml of hypochlorite solutions are added. The tubes are placed in boiling water for 20 minutes, and a positive test is indicated by a blue color. Another test is the pyrazinamide test. Here the enzyme pyrazinamidase hydrolyzes pyrazinamide to ammonia and pyrazionic acid, which is detected by adding ferric ammonium sulfate. The positive reaction is indicated by a pink color band, and the Mycobacterium tuberculosis gives positive for this test, whereas Mycobacterium bovis is negative for this test. Many antigens have been identified in Mycobacterium. Group specificity is due to polysaccharide and type specificity is due to protein antigens. The cell wall antigens consist of the peptidoglycan layer, arabinogalactan layer, mycolic acid layer, and the mycocytes. And the cytoplasmic antigens consist of the protein antigens. The mycolic acid layer in the cell wall antigens provide the following property such as the difficulty to stain with other stains, difficulty to phagocytose, and help in the intracellular survival, delayed hypersensitivity, slow growth, and the resistance to heat and chemical disinfectants. There are also various typing methods for the Mycobacterium tuberculosis that includes phenotypic methods and molecular typing methods. The phenotypic methods include the bacteriophage typing, and based on that, the tubercle bacilli has been classified into 4 phages, mainly A, B, C, and an intermediate between A and B, and it is designated as I. So it's A, B, C, and I. The next one is the bacteriocin typing. In this, it is typed by means of bacteriocin produced by the rapidly growing Mycobacteria. And the molecular typing includes RFLP typing, where the restriction endonuclease treatment yield nucleic acid fragments of varying length and the pattern of which are strain specific. And there is spacer oligotyping based on polymorphism in the direct repeat locus. And these all typing methods help in the epidemiological study of the diseases. Mycobacterium tuberculosis causes natural infection in humans and other primates, dogs, and other animals in close contact with the humans. It is highly infectious for guinea pigs and hamsters. Mycobacterium bovis is more pathogenic for animals. It causes tuberculosis in cattle, carnivores, dogs and cats, swine, parrots, and other birds of prey. It is moderately pathogenic for rats. African strains or Mycobacterium africanum was isolated from some parts of South Africa, showing intermediate properties between humans and bovine types. Whereas Asian type of tubercle bacilli was isolated from South India, where of low virulence to guinea pigs was found.